Hey guys, Shreyans here. Today we are going to be learning about cell division. Now, cell division isn't just any division, it's division of the cells. Now, you're probably asking yourself, why do we need cell division? Well, it's pretty straightforward, guys. We need cell division to reproduce, heal injuries, repair dead cells, and for other reasons as well. Now, according to every informational article, Walter Fleming was actually one of the first to devote his time to the study of chromosomes. Fleming was the first to detail the chromosomal movements in the process of mitosis. Now, in 1879, Fleming used a byproduct of coal tar to stain cells of salamander embryos. He was able to visualize the thread-like materials as the cells divided. He described the whole process in his book called Zellsubstanz. Now, we have some background info on cell division, so let's get right into how it works. Now, cell division is pretty self-explanatory if you know the basic information about it. It's the process that increases the number of somatic body cells. Now, the process consists of mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. For example, if you have a cut or scab, cell division will repair the injury and basically heal you. Mitosis is involved during embryonic movement, development, growth, tissue repair, and asexual reproduction. Now, before a cell divides, it needs to undergo preparation for division. This phase is called interphase, which can take a long period of time. Now, interphase is divided into 3 stages. First stage is the G1, which is the growth phase, number 1. Next is the S synthesis phase, 3. Then after that is the G2 growth phase number 2. Now, make sure that you know interphase takes place before the cell undergoes mitosis and divides. In early prophase, which is the first step of cell division, the cell starts to break down some structures and build others up, setting the stage for the division of the chromosomes. Now, the mitotic spindle begins to form, which is a structure made of microtubules, and its job is to organize the chromosomes and move them around during mitosis. The spindle grows between the centrosomes as they move apart. The nucleolus also disappears in this phase. Now, in late prophase, the mitotic spindle begins to capture and organize the chromosomes. The chromosomes have finished condensing, so they are very compact. Then, the nuclear envelope breaks down, releasing the chromosomes. At the end, the mitotic spindle grows more and some of the microtubules start to take a hold of the chromosomes. Now, after prophase, metaphase occurs, where the spindle has captured all of the chromosomes and lined them up in the middle of the cell. Now, before the cell proceeds to anaphase, it will make sure that all the chromosomes are at the metaphase plate with their connectors correctly attached to the microtubules. This is called the spindle checkpoint and helps make sure that the sister chromatids will split evenly between the 2 daughter cells. Now, in anaphase, the sister chromatids separate from each other and are pulled towards opposite ends of the cell. The protein that holds the sister chromatids together is also broken down, allowing them to separate. Each is now its own chromosome, and each pair is pulled towards opposite ends of the cell. The processes are conducted by a motor proteins, which are molecular machines that can move along microtubule tracks and carry cargo. Not actual cargo, but cargo. Now, the next step in cellular division is telophase. During this step, the cell is nearly done dividing and it starts to re-establish its normal structures as cytokinesis, aka the division of the cell's contents, takes place, which will be talked about later on. Now, during telophase, the mitotic spindle is broken down into its building blocks. Then 2 new nuclei form, one for each set of chromosomes. Nuclear membranes and nucleoli reappear and the chromosomes begin to decondense and return to their stingy form. Now, finally, is the step we have all been waiting for, cytokinesis. Now, during cytokinesis, the cytoplasm divides to form 2 new cells, while overlapping with the final stages of mitosis. It may start during anaphase or telophase, depending on the cell, and will finish shortly after telophase. Now, basically, after cytokinesis, we're pretty much done. When cytokinesis finishes, we are left with 2 new cells, each with a complete set of chromosomes identical to those of their mother cell. And that is it, guys. Thank you for watching, and hopefully you have a nice day.